Good morning, students. So today we are just studying about the basic terminology in microbiology. So before going to this introductions, we must know about some basic terminology related to the microbiology. Now, first is disinfection. So, what is disinfection? So, it is the process to kill the microorganisms with the help of the chemical disinfectant. It means you can, with the help of the chemical disinfectant, you can kill the microorganism, but you will not able to kill the bacterial spores. So, means that your articles, that your instruments, that will be clean, but you will not be able to completely sterilize. The second is sterilization. So sterilization is it is the process to which you will be able to kill the microorganisms including the spores. Okay, so it means the main difference in the disinfection and sterilization is means in the disinfections you will be able to stop the bacteria, but you will be not able to completely killing the bacterial spore. But in the sterilizations you will be able to kill the microorganism including the bacterial spores. Now next term is that is bactericidal and the bacteriostatic. Again we are having these two terms. So basically we are having the difference in these two terms is the bactericidal is it is a chemical and the biological substance which kill the bacteria. Okay, the same thing this is the substance through which we can able to kill the bacteria including the bacterial spores. But if we are talking about the bacteriostatic, so means it is a substance which stops the bacteria from the reproducing. It is only stopping the bacteria from the reproducing. It is not able to completely killing the bacteria. So this is the basic difference in the bactericidal and the bacteriostatic. Now next term is we are having that is pathogen. So pathogen and the non-pathogen. So pathogen is basically related to that microorganisms which is able to cause the disease inside the body, but the bacteria which is not able to cause the disease inside the body, that type of bacteria is known as non-pathogen. So pathogen which is causing the disease inside the body and the non-pathogen which is not able to cause the. Again, we are having these two terms, that is aerobic and the anaerobic. Aerobic means the bacteria which is growing in the presence of the oxygen, and the bacteria who are growing in the absence of oxygen is known as anaerobic. Now the next term we are having, that is virulence. So the virulence is the basically it is the power of producing a disease by the microorganism. Means the bacteria producing the disease power is known as a virulence. Means whenever the bacteria is entered in your body, so basically it depends on the power of producing disease. So some bacteria we are having a less virulence power, and some bacteria is having a more virulence power. So the virulence is the power of producing disease is known as a virulence. Now the next term we come to, that is attenuation. So attenuation is it is the process of weakening the power of producing disease. So it means the power of producing the disease, that is virulence. So we can weak the virulence power of the bacteria. Okay, so this is the process which helping in the weakening and reducing the power of producing disease, that is virulence power, is known as a attenuation. So basically for example we can give, like we are using the antibiotic therapy, we are using some vaccines, we are using some drugs when the patient is suffering from any disease condition. So it means with the help of that antibiotic we are reducing the power of disease, we are reducing the power of producing disease. So this attenuation means this process, this antibiotic, whatever the process which we are using, which is basically included the antibiotic therapy, the drugs which we are using, so that are helping in relieving the sign and symptoms. So this sign and symptom will be relieved by reducing the power of producing disease. So this all process is known as a attenuation. Okay, so next term we are going to the base. Again we are having some basic terms, that is sepsis and asepsis. Okay, so when the person is having an infection, so means the presence of infections in the body is known as a sepsis. When there is an absence of infection, so that is known as a asepsis. So the sepsis means presence of infection and asepsis means absence of infection. So now we come to next terminology in microbiology. First is that is invasion. So what is the meaning of invasion? Invasion means this is a basically entry of the microorganisms into our body through any medium. Okay, means whenever any microorganism has entered in your body through any medium, it can be through your nose, it can be through your mouth, it can be through your eyes, it can be through any medium, through with the help of any medium. When any microorganism has entered in your body, only just entered in your body, so that is known as a invasion. So we can say it is the invasion of the microorganism. Clear? Now we come to the next term is that is infection. So infection is, so we can say infection, it is the invasion of organism into the body tissue which lead to multiplication of bacteria and cause disease. It means, like we have learned first term, that is invasion, means in that only the microorganism has entered in your body. But what happen in the infection, after the invasion of microorganism into your body, it will start multiplication. The bacteria will be proliferate, the number of bacteria will be reproduced from a single bacteria, and after the multiplication of bacteria, when it will cause the disease and it will show the sign and symptoms inside the body, so that indicate that invaded bacteria has reproduced in your body and has caused disease in your body. So this is known as a infection. So infection the basically mean producing a disease conditions after entering in your body, which is manifested by some sign and symptoms. So that is known as a infection. Okay, now next is we come to that is prognosis term. So prognosis means we can say it is a predicted course of disease. Okay, or we can say recovery of a patient from a disease. So basically the prognosis, it is a recovery rate of a patient from any disease conditions. Okay, like we are saying basically we use that term like the patient example, like the patient is suffering from any cancer, that patient is suffering from any other disease conditions. So the physician used to tell ki, the patient having a 10% recovery rate, 90% recovery rate. So means the basically the prognosis, what is the prognosis of the disease? So basically recovery of the patient from any disease conditions is known as a prognosis. So that can be predicted in a percentage or that can be predicted in a days. So this is known as a prognosis. Now next we come to term the basically related to the experiment, that we are doing experiments. So we have divided some terms, that is in vitro and the in vivo. So in vitro, it is the experiment which is done in an artificial environment, like any experiment which we are doing in the lab, in which we are using some artificial environment. So that type of experiment is known as a in vitro. And the experiment which we are doing in the living beings, like we are using some rats, we are using human, we are using some horse, so we are doing some experiment in the living being, that is known as a in vivo. So basically we can take the example, like if we want to apply, we want to try any medicines, any vaccines in a patient, in person. So before going to the human being, first we use any animal to do the experiment. So that type of experiment is known as a in vivo, because we are doing the experiment in the living beings. Now the next term is we are having, that is incubation period. Incubation period means we can say it is a time period between the entry of microorganism up to the appearance of sign and symptoms. It means when the bacteria has entered in your body. So this will be your first day of entry of bacteria, and the time period when there is the presence of sign and symptoms. Okay, so means this is the time period between when the bacteria has entered up to the sign and symptoms is present inside the body. So this time period in between this organism is known as a incubation period. Okay, so that is known as a incubation period. So now we are giving some examples, like this bacteria is having an incubation period for a 14 days and for a 28 days, means within 14 days means it after the entering into the body it will show the sign and symptoms with after 14 days. So means the 14 days is the incubation period. So we will say this is the incubation period of showing and sign and symptoms. Next we are having quarantine period. So quarantine period is like recently we are very familiar this term, that is quarantine and the isolations like in this pandemic. So the basic meaning of the quarantine means it is the separation of a person. Okay, it is the separation of a person or the people from the other person, means to whom we are separating. This is restricted the people who have been exposed to any contagious disease, means we are not sure that that person is suffering from any disease, any communicable disease, but we are known that he has exposed, but it is not confirmed it is having, the person is having the confirmatory diagnosis. So means before, so it means for the prevention of a disease conditions to the other persons, we are separating out the person. So we separating out the persons from the other persons to prevent the infections from that person to the healthy person. So that is known as a quarantine period. Okay, now next term is we are having, that is isolation. So isolation means we are separating the sick people. Means sick people means we are known that that person is suffering from this specific disease condition. Now the person is confirmatory diagnosis that this person is having this disease. So that's why we are separating this sick people from the healthy person. So that term is known as a isolation. Okay, so we can identify the difference in the quarantine and the isolation. Isolation means we are separating the confirmatory person, means sick people, separating the sick people from the healthy person. But in the quarantine we are not separating the sick people, means we are separating that persons who have been exposed but the person is not having any disease conditions. Okay, so that is known as a quarantine period and the isolation period. Now we come to the next term, that is immunity. So this is a very basic terminology. Immunity means it is a resistance of a host to pathogen and their toxic product, means it is the immunity of a person to fight against the foreign particles. Okay, so that is the immunity, means it depends on individuality. And the basic, like we are having a WBC, so this is a basic cells which is helping in the fighting against the any foreign particles inside the body. So that is known as a immunity, means your body is having a resistance to fight against the any foreign particle, that is known as a immunity. Next we are having, that is prophylaxis. Prophylaxis means it is intended to prevent the disease, means the disease means that before the occurrence of any disease conditions we are taking any preventive action, we are taking any preventive measure. So that is known as a prophylaxis. Like we can take an example of a immunization schedule. This is a very common example. Like in the immunization schedule we are giving vaccines to the baby. Up to the 5 year we are giving immunization. So means like we are giving the BCG injection, we are giving the DPT injection. So basic means is the baby is not having this disease condition, but before the occurrence of this disease we are giving that vaccines for the prevention, so that in the future, in the further conditions, if the baby has exposed to that type of microorganism, so that disease will not occur in the baby. So means we are taking the precautions, we are taking the preventions before the occurrence of disease condition. That type of thing is known as a prophylaxis. Next we are having, that is exogenous infection. So exogenous means infection, already we learned. Infection means it is a multiplication of bacteria which is causing sign and symptom in the body. But if that infection has occurred from outside of your body, so that is known as a exogenous. And endogenous infection is when this infection originating itself inside in your body. It is not coming from outside of the body, it is coming from your body only. So that is known as a endogenous infection. Okay, now next we come to, that is fulminating infection. So fulminating infection means the infections which occurs suddenly. Okay, means the sign and symptoms which shows very for a short duration and suddenly sign and symptoms come. So that is known as a fulminating infection. So now we come to next term, that is a culture media. So culture media means it is a liquid gel or a gel substance containing the nutrients in which microorganisms are growing. So it means it is a basically a gel substance which is used to grow the bacteria in an artificial medium. So that is known as a culture media. Like example for the culture media, we are having agar gel. We are using agar gel to grow the bacteria artificially. Next we come to inoculation. So inoculation means to introduce the microorganism into a culture media at a specific temperature and the pH to grow a number of colonies of the bacteria. It means from the specimen, from the person's specimen, we are isolating the bacteria, and after the isolation we are introducing the bacteria into that artificial medium. So that process from isolating the bacteria from the specimen up to the introducing into the microorganism is known as inoculation, which is very common while identifying any bacterial structure of the person and while doing any experiment. So that this term is very commonly used. So I think it is clear what is inoculation. Now next term we are having, that is fumigation. So fumigations, like before in the first slide, very beginning slide, we have learned what is sterilization. So basically in the sterilizations we are using some chemical substance, but if we are using any fumes or vapors to sterilize the things, so that is known as a fumigation. The basic meaning is we are providing some fumes and vapors at a high pressure to sterilize the things, to prevent the multiplications of the bacteria and also killing the bacterial spores. So for example, if we this is fumigation, this process is very basically used in the hospital, like in the ICU, in the operation theater. If we want to sterilize that ICU area, if we want to sterilize the ICU room or the operation theater, we want to sterilize, so we are using this process, that is fumigation. Okay. So that was about the old terminologies. So this is a reference from where I have taken this terminology. I think it is clear. These all terminology are clear to all of you. So we just studied about the basic terminology which was used in a microbiology. Thank you.